I'm a postdoc at the Department of Insulin Biology at Novo Nordisk, and for the past year and a half, I've been working in the lab of Mark Hillestein at UC Berkeley in California. And there I have learned to use heavy water labeling, which is a technique I can say I have not heard about anyone using it in Denmark before. And we used it to follow lipid synthesis and turnover in the setting of insulin therapy. And what I would like to start out with is, first of all, why do we even bother about measuring insulin's effects on lipids? And then I want to talk a little bit about how we do this heavy water labeling, and then end up with some of the results that I found. But to start out with insulin, it's not all about glucose. If we take a closer look at what happens when insulin reaches the liver, up here, the main effect is that insulin sort of makes the liver clear glucose from the blood. So we have hyperglycemia here, which is relieved. And it's done by insulin inhibiting the endogenous hepatic production of glucose and by the liver taking up all of this excess glucose. All of this glucose needs to go somewhere. And insulin also stimulates the liver to produce fatty acids from all these glucose molecules. Those are incorporated into triglycerides and exported in the form of VLDL particles to go to the fat depots for storage. So it's basically an efficient way of storing all of this excess energy. But when it comes to insulin resistance, there seems to be an imbalance in these pathways. So what is believed to take place is that the pathways down here that affects the glucose metabolism, they're much more resistant than the lipid pathways. And in the treatment of type 2 diabetes, where these patients are usually insulin resistant, we have to give them rather large doses of insulin in order to overcome the resistance in the glucose metabolism. So there is a concern that what might happen is that we hyperstimulate the lipid pathways. So to keep down the glucose, we're inducing a lot of lipid synthesis. And in the setting of cardiovascular disease, the concern is that we might affect the risk of this disease with a increased production of lipids, and even more so with lipids that might end up in the right places, in the wrong places, of course. So this is something that we might need to consider in the development of future diabetes or insulin therapies. But in order to do so, we need to be able to measure what's going on with lipid metabolism. And lipids are a little bit hard to get a hold of, but Mark Hillestein, that I've been working with, he's been in this field for more than 30 years, doing a lot of metabolism studies, and especially with lipids. So what he's doing at his lab is that he uses heavy water labeling, and chemically speaking, heavy water is the exactly same as regular water. The only difference is that instead of hydrogen, it contains deuterium, which has an extra neutron in the nucleus, which makes it 1 mass unit heavier. So the way that we do these experiments is depicted here. So we have groups of animals that are treated differently, and we give them all an injection of heavy water. And this is non-toxic. It doesn't really have any known effects, except there is one side effect that's actually kind of freaked me out in the beginning, because the animals get very dizzy. And this is believed to be due to the equilibration across the balance nerve. And the first time I injected my batch of mice, I seriously thought they were all going to die. They curled up in a corner of the cage and wouldn't move, but they were so dizzy, and 20 minutes later they were running around like nothing happened. But what happens is that this water, the heavy water, mixes with the remaining body water pool. We aim for around a 5% enrichment. And then by the end of the study, or during, or in the presence of heavy water, in the synthesis of biomolecules such as lipid, DNA, proteins, there'll be an enzymatic transfer of hydrogen from water onto these molecules. This means that when we have heavy water present, there will be deuterium attached to all of these molecules. And by the end of the study, when we take down the animals, we can take out the tissues of interest, isolate the molecules that we want to look at by mass spectroscopy, and we can see how much labeling has been taking place, which is a measure of how much synthesis has actually been going on in the labeling period. And for that, Mark Hillestein has developed some algorithms which we can use for calculating these things. So we'll move on to talk a little bit about some of the studies that I did while I was there. I did one study where we wanted to see what is the effect of chronic insulin treatment on the adipose depot. So we chose to use some mice that were not insulin resistant to start it in sort of an insulin sensitive model, which is just regular C75 BL6 mice. And once there, we started treating them with insulin, as shown with the blue curves. We saw that the glucose levels went down, which is where we can see that the insulin is actually working. We also saw that the animals actually gained weight. They gained significant weight with the high dose of insulin, but we did not find that they actually ate more. So we looked into the adipose depots. And here I dissected out the subcutaneous depot, which is situated right here, right underneath the skin, and also the gonadal depot. These were female mice, and it's here right next to the ovaries. And I found that there was a significant increase in weight after 1 week of treatment in the subcutaneous depot as compared to the controls. I saw the same picture in the gonadal depot, but it was non-significant due to the variation. I then looked into the mechanism for this fat accumulation. And the first curve up here, or the first graph, is basically the exact same as before. I just subtracted all my control values to see how much more triglyceride did my insulin-treated mice accumulate as compared to the control mice. So we can see that the insulin-treated mice accumulated approximately 25 milligrams more of triglycerides per day. And we saw a tendency for the same increase in the gonadal depots, but again, not significant. Then with the heavy water labeling, I used this to calculate, well, of all this triglycerides that the mice accumulated, how much of this was newly synthesized. And I did that by looking into the glycerol moiety of the triglycerides to see how much labeling has there been going on. Whenever the liver or the adipose tissues make triglyceride, they usually synthesize a new glycerol that they used for that. And we can see that in the subcutaneous depot, we saw that mostly all of the fat that accumulated was newly synthesized, but we didn't really see anything of that in the gonadal depot. So that leaves us with the question, how can the gonadal depot increase in size but without having any new fat in there? And the answer to this might come from lipolysis. So here, insulin also inhibits the export of fat out of the fat depots by inhibiting lipolysis. And down here is a graph of how much lipolysis there is as compared to the control group. And we can see that in the subcutaneous depot there is hardly any difference in lipolysis rates, whereas there is a less lipolysis going on in the gonadal depots. So without heavy water labeling, we're only able to see that there was an accumulation of fat mass, but with heavy water labeling we could distinguish this between two different mechanisms. I also did a study in diabetic rats, the so-called Zucker diabetic fatty rat, which has a non-responsive leptin receptor. So it eats a lot, it's very hyperphagic. And around 12 weeks of age, they turn diabetic. And so I treated those with insulin again, chronic insulin infusion, by implanting a little pump under their skin. And down here, the light green line is the lean control animals, they're normal glycemic, and up here are the diabetic animals, which were grossly hyperglycemic by the beginning of the study. But when I started treating them with insulin, their glucose levels went down relatively fast to be in the normal glycemic level. The purpose of this study was to look at the hepatic lipid production, and in order to do so we labeled for only 2 hours because the turnover of lipids in the liver is very fast. So if I labeled for too long, I would just see 100% of labeling in all my groups and I wouldn't be able to tell the difference. So we looked into the amount of triglycerides that were present in the plasma. That is a sort of a measure of how much lipids are exported in the form of VLDL particles. And also I looked at the total amount of palmitate in the livers. And when I did that, I didn't really see anything going on. I could see there was a large difference to when the animals were lean controls as to when they were diabetic. And I saw that in both of my parameters also here. But it didn't really seem to matter if we treated these animals with insulin or not, so our hypothesis did not seem to fit with what we saw. But then I started to look into how many of these lipids are actually new. So when I looked at this thing called fractional palmitate synthesis, it's basically a measure of all the palmitates that are incorporated in the triglycerides, how many of these palmitates has been newly synthesized within the past 2 hours. I could see there was a huge difference. With the lean controls and the untreated diabetics, there was only around 20% of these palmitates being new, whereas with the insulin-treated animals, this was up around 80%. And I saw the same picture with the amount of palmitate in the liver. Again, of course, less palmitate in the lean controls. They were not steatotic, but a slight increase when they became diabetic, and then a huge increase when we started treating with insulin. So we could actually tell that as soon as we started insulin treatment, we really got a massive response by de novo lipogenesis. And I would like to thank my supervisors / colleagues, Mark Hellerstein, Thomas, Christian, and Grit from Novo Nordisk, and the STAR program from Novo Nordisk, which has sponsored my postdoc. And then finally, as some of you may know, Berkeley is a place where a lot of old hippies still reside. And there is this saying that Berkeley is the place where the nuts go chasing the squirrel. And from what I've seen, there might definitely be something about it. But thank you for your attention, and let me know if you have any questions.